Okay, I'm gonna go ahead and get started on the lecture for today. Looks like we do have somebody here with us, so if you have any questions, you're welcome to add them to the chat. I've got the little chat box open, so I'll try to get to those as I can. This is going to be the Chapter 4 lecture video. I'm just going to be doing this one video, and so it might be a little bit longer than some of the other ones. It might feel a little fast. So if you need to watch this in multiple parts, don't try to just do it all at once, because that might be a little overwhelming because there's going to be a lot of information coming at you. This is starting our energy chapter. So chapters 4, 5, and 6 all kind of go together with unit 2. We have chapter 4 is just going to introduce us to the basic concept of energy, the different types of energy that we see in biological systems. Chapter 5 is photosynthesis. Chapter 6 is cellular respiration. And so we're going to be kind of taking these ideas of, well, what is energy? How do organisms create and make their own and use energy? And then as we get into chapters 7, 8, 9, that's all about DNA, cell reactions, protein synthesis, and those kind of things. So let's jump into chapter 4 here. So all cells capture and use energy. All cells have to use energy. All organisms have to use energy, even if they appear to hibernate, even when they're sleeping. There's not a time when an organism stops using energy. We can slow down our energy use. Some organisms can. They can regulate their metabolism. I have a pet ball python. He's about 8 years old, and he can go for months without eating because he can slow down his metabolism. He can slow down his energy needs, but he still has needs. He still has to have energetic reactions happening. Lots of different reasons that we would use energy, right? For cell transport, for synthesizing proteins, for removing toxins from our blood, all those different things that we might need to use, growth and repair, right? All those reasons that we would use energy. And so specifically, energy is the ability to do work. We can quantify it in the terms of energy, but energy is the ability to actually do something to make a change. And we're going to see that in a lot of these things, energy comes down to electrons. And energy comes down to being able to change the shape of things, breaking and forming bonds. So energy can be converted. That's one of the natural laws of thermodynamics. If you've taken some chemistry, we've talked about the laws of thermodynamics. You may have heard that one about energy can neither be created nor destroyed. Everything that we have, all the energy that we're using, comes from things that are billions of years old. We're not making new energy when you turn on your lights, or when you make a fire, or when you go and run a marathon. That's not new energy that's being created. It's being transferred from one source to another. The energy when you go to run a marathon, right? That doesn't just come out of nowhere. You had to eat something. You probably had breakfast. You had a pre-workout. You're having a snack in the middle of the marathon. You're consuming something. You're consuming food, and your body is converting that into a usable form of energy. We have to consume ours. We have to eat food to be able to make energy. Other organisms can make their own energy. They can make their own food through photosynthesis. And so we're going to look at some of that, some of those changes. Here you can see kind of a general energy diagram. Sun is the original source of energy in our universe. That is what provides that initial burst of energy for us. The fusion reactions of the sun put off all of this heat, all of this light energy that can be captured by photosynthetic organisms, and they can convert that into sugar. So they're taking this sunlight, this light energy, and converting it into this physical form, this sugar energy. And then we can eat those plants, right? Organisms can go and then eat those plants, eat the fruit, eat the vegetables, whatever it may be, eat smaller organisms. And through cellular respiration, we can then release this energy that's been stored in the sugar. So you start off with this kind of light energy that gets converted to sugars. Sugars then can be eaten and released through cellular respiration to produce more energy. And so it's this kind of cycle. So we're going to look at each of these steps in this chapter. And then again, chapter 5 is all about photosynthesis. Chapter 6 is all about cellular respiration. So we'll get really in depth into those reactions in those chapters. Something that we need to look at, though, is that at each of these points, you see that you have heat energy being lost. Even though energy can't be created or destroyed, our reactions aren't perfect. There's always going to be some loss of energy in the form of heat. That's why we are warm-blooded organisms. That's why we produce body heat. Body heat comes from the heat that is lost during cellular respiration. Your muscles are constantly working, constantly creating energy, burning calories and creating energy, and in the process, they produce heat, and that's what keeps our bodies nice and warm. That's what keeps us at that body temperature, stable temperature. When you go to drive your car, if you know you're pulling up to somebody's house and you're like, oh, I wonder if they're home or not, and you can still hear their car kind of like crinkling and popping from where all the metal is kind of starting to cool down. You're like, oh yeah, they were just driving the car. You can feel the heat coming off of their car, right? So heat gets lost in these energetic reactions, and some cells, some places, it's able to. It's always a waste product. It's not usable, but we can find ways to harness it in a way, right? Like body temperature. Or we can use the heat to keep us at an appropriate temperature for reactions to take place, for life to take place, for us to exist. So while heat can't be used specifically as an energy source, that would be something like a perpetual motion machine, right? Where if you had a battery and the battery powered a wheel that was spinning, and as the wheel spins, it's going to put off heat. And if you could somehow recharge that battery from the heat that was being put off from that spinning wheel, think about like a car, right? As a car is turning, if you could somehow get the heat from that car engine to then recharge the battery, that would be a really incredible, I mean, that's a reusable energy right there. We can't do that. We don't have that kind of power. We don't have that kind of technology. So heat is just lost as this energetic form. What I want you to think about, because energy is the ability to do work. It's the ability to do something. It can be motion, right? Like doing something, it's an action, you're changing something. In that, the idea of action, it's movement, right? Like if you apply energy to something, you then think about it moving. Well, heat is essentially just increasing the movement of molecules. What we experience as heat when, like, right, today it's 80 something degrees outside and it's been so nice and cool, and so you might feel heat, right? What's really happening in your body though, as your body gets warmer, or as you touch something and something feels warm, what's happening is that the molecules are speeding up. They're actually interacting and hitting each other more. Heat is a physical reaction of molecules to speed up the action of their movement. Okay, think about water boiling. When you go to boil a pot of water, you're applying energy to it, you're applying fire to it, and what's happening is that it's causing those water molecules to hit up against each other more. And as they hit up against each other, they can break and form bonds, and so that is a way that heat can then be kind of harnessed, or can be contributing to systems, is that heat is going to increase what we call the entropy, the chaos of the system. And as you increase the entropy, you increase the rate of reactions. So heat increases entropy, increased reaction rate. And so heat itself can't be directly used as an energy source, but it can help to facilitate reactions. That's why we say we need to have body temperature for us to maintain function. So energy does have different forms. Energy can be seen in different ways. Like I said, energy, if you think about it, you apply energy to something, you think about maybe it moving, right? You're kind of energizing it, you're activating it, so it's a moving thing. We call that kinetic energy. When something actually moves, when you apply energy, or something is moving, it's changing, right? That is kinetic energy. So right now I am using kinetic energy. I'm talking, I'm gesturing, right? I'm clicking the slides. I'm using my kinetic energy. You are probably using your potential energy. You're probably sitting there watching this, listening to me, whatever it may be. You're not really doing anything. You're just kind of sitting there absorbing it, right? Your brain's working, but your body really isn't having to do much. And so you're kind of converting, you're using your potential energy. You have the potential to do something. The common classic example, right? These two right here. When you have a bike, when you're kind of perched on the top of a hill, you have your maximum potential energy, right? If you were to roll, if you were to move, oh yeah, you're now releasing that potential energy, right? In this sense, it's like, oh yeah, there's really nothing there. But let's say that you attach some kind of pulley string to yourself that was then attached to another machine, and as you rolled down the hill, you pulled and you started that machine's motor, right? So then you're harnessing your potential energy. And so there's always these two forms of energy. You have potential is one kind, and kinetic is another. A compressed spring, right? The spring at fully compressed, this has high potential energy. When you let that spring go, boom, that potential energy is being converted into kinetic energy. This doesn't happen in an instant, right? It's not like you start up here at the top of the hill and then magically, magically or instantly you are at the bottom of the hill. No, you have time where you're actually having to coast down. You're actually having to move down the hill. Same thing with the spring. It goes from fully compressed to fully extended. Well, it takes time for that to happen. So as this is happening, as you're going down the hill, as the spring is being sprung, potential is being converted to kinetic. It's a process, right? We see it happening immediately, right? But if someone were on this hill and they were to stop halfway, well, they still have some potential energy, right? They still have energy that's going to take them down the rest of that way. So they haven't used all of it up. And so we can convert potential energy into kinetic or kinetic into potential. We can do it both ways. And it can be a process. You can convert some of the potential into kinetic and then you can reserve it. That's like if you were to go sprint. You're trying to run away from something. When you go and sprint, you don't use all of the stored energy that you have in one moment. You use some of it, and then you start to notice, oh, I'm getting a little winded. I don't have quite as much energy. I need to slow down a little bit and conserve my energy, right? So we can convert some of our potential. It doesn't have to be all of it converted at once. So you might be wondering, well, what is, we talk about, I just said converting our potential energy. What does that mean? Are we breaking down like our muscles? Are we breaking down our organs? Like how do we get that energy released? Chemical bonds. Chemical bonds are our source of energy. So a chemical bond, in this case, we're looking at a glucose molecule. It is a storing energy, right? It's holding on to energy inside of these bonds. So, if you think about when someone is low in energy or they're feeling a little sluggish, they kind of say, "Oh, well, you need to get some sugar in you. Your blood sugar is low. Let's get your blood sugar up." The reason that we talk about blood sugar, and specifically the sugar is glucose, the reason that we talk about that is because your blood sugar is that sugar glucose molecule is your source of kinetic of potential energy. Our bodies can break open glucose molecules through cellular respiration, release those, and release that energy that's stored within. And when they do that, that energy release, that's not heat being released, right? It's a different kind. So there is some heat that gets lost. But what you're seeing here is actually stored energy. And so simply by breaking these bonds near other things, near reactions that you want to happen, you can fuel that reaction. We kind of are used to like energy, like I have, let's see if you'll be able to see it. Like I've got my mouse pad kind of like plugged in, right? Like that's how we're used to energy kind of things happening, like a battery. We're used to this kind of very much like direct attachment. But really when you're looking at a molecular level, things are so small, things are so close, as long as they happen near each other, they're happening close enough to each other that when you go to break open this molecule, the thing that it's trying to fuel is right there beside it. So it's going to absorb that energy really quickly. It's kind of like if you go to shock someone, like if you're jumping on a trampoline and you go to touch the edge of it, or if you go to touch someone else, you don't actually have to touch that person. You can just get kind of close and the energy can jump the rest of the way. That's what's happening when we break bonds. We talk about breaking bonds to fuel a reaction. Okay, so chemical bonds are potential energy. When you make bonds, you are storing energy. So in photosynthesis, when photosynthesis is taking light and using light energy to make sugar, that is a storing energy. They're storing light energy in a sugar molecule. And then through cellular respiration, we can break open that sugar molecule, convert the potential into kinetic, and release that energy and provide energy to fuel reactions. So there's two different types of reactions then when we're talking about either building chemicals or breaking chemicals down. Altogether, it's called metabolism. That's probably a word you're familiar with. Metabolism refers to all of the chemical reactions in your body, all of the breaking and all of the making. So this chapter really kind of, we try to organize things. So if you look at the soft chalk, we kind of talk about how you want to group and categorize these reactions and think about them in terms of, well, is this a kinetic reaction or a potential energy reaction? Is this going to be a reaction where we're breaking something or making something? Yeah. So chemical reactions rearrange atoms. That's what chemical reactions are about. You have your reactants on one side and then products on the other. Reactions can either be making things or breaking things. You can build complex molecules out of simple ones. So here you can see you have a couple of these smaller little monomers, these little small building blocks. You put energy into it, you're storing energy in the system to make bigger things. Okay, you're forming complex molecules. We call these anabolic, anabolism. Sorry, so that emphasis was weird. Anabolism. So anabolic reactions, you are building things. Breaking molecules, you start with this large molecule, and as you break that molecule apart, you release energy. This is called catabolic, so catabolism. So you have anabolism, where you're making, catabolism, where you're breaking, and together those are metabolism. So metabolism is anabolism and catabolism, right? Making and breaking all together are metabolic reactions. So when someone talks about their metabolism slowing down, as you age, your metabolism slows down. What they're talking about is that their body's rate of reactions is slowing down. You don't build structures like you used to, right? When you're younger, when you're growing, you have a lot of hormones pumping through you, and so you are doing a lot of growth and a lot of repair, a lot of breaking and making reactions, metabolism reactions. As we age, we don't really repair quite as much. We don't have these reactions quite as fast, and so our metabolism is slowing down. So in these reactions, when you're building complex molecules where you're putting energy into a system, right, we're storing energy. So this is creating potential energy. We are creating molecules that have potential energy, whereas in this one, we're releasing that energy. So this is kinetic reactions. You're releasing that energy, you're moving it out, and so that is a kinetic reaction that we're seeing. So catabolism is kinetic, anabolism is potential energy. We can also talk about it in terms of, so let me go back real quick, sorry. These words we're talking about breaking and making. This is looking at the physical arrangement. So with all these next kind of terms that we're going to be using, it's all about how energy is being exchanged, but we're looking at it from a different viewpoint. And that's something that biologists and scientists love to do, is they like to take one thing and then look at it from different angles. So it's all going to be, all these next slides are all about just reactions. You're either forming bonds or you're breaking bonds. But in that process, you can look at it from different ways. So with metabolism, with anabolic and catabolic reactions, we're looking at the structural changes. When you talk about a catabolic reaction, you're talking about the physical structure being broken apart. An anabolic reaction, you're talking about the physical act of bringing things together. So you're talking about the physical structure itself. These next terms, endergonic and exergonic, this isn't actually looking at like the structural change of, oh, things are coming together, things are coming apart. This is looking at how the energy is being moved. Where is the energy going? So endergonic describes reactions where you're putting energy into a system. When you put energy into a system, are you building things? Is that an anabolic reaction? Yes. But again, we want to have different terms to look at these from different angles, because you never know what your specialty might be, right? When you go and you specialize in your biology career, you never know what you might be, where you might need to be looking at things from. So we can talk about anabolic and catabolic. Anabolic, building things, catabolic, breaking things down. Endergonic describes the fact that we're putting energy into these reactions. So you can see here you have these reactants down here at this lower potential energy on this side. As the reaction progresses, energy is added and it lifts them into this higher potential energy state, right? This is like sunlight, right? You put sunlight into a system, energy required, sunlight going into a system to make a glucose molecule. So this is how photosynthesis occurs, right? You take these smaller monomers to make this larger polymer, and the energy that has to be supplied, that comes from sunlight. So this is an endergonic reaction. Ender, into, right? Ender, endo, into, right? Remember that prefix. Exergonic reactions, exit, so energy is leaving the system. So you start with high energy products like a glucose molecule, and through cellular respiration, you can break those into their smaller products. So this is a catabolic reaction. Catabolic reactions, though, release energy, and so we describe that process as being exergonic. So if you want to talk about the energy movement of reactions, you can talk about an endergonic reaction where you're putting energy into it, or an exergonic reaction where energy is being released from it. Exergonic reactions are what fuel biological processes. Right? Okay, let's keep going. We can also talk about the electron transfer. So when we talk about making and breaking bonds, bonds are electrons. If you remember that from chapter 2 or chapter 3, whichever chapter that was, covalent bonds, ionic bonds, you're talking about what's happening to the electrons. And so specifically, we can then talk about redox reactions or oxidation reduction reactions. So anabolic, catabolic, you're actually talking about the structural change, taking monomers and making polymers, polymers into monomers, whatever it may be. Endergonic, exergonic, you're talking about how the energy is moving. Is energy going in, endergonic? Is energy being released, exergonic? Oxidation reduction reactions, this is specifically talking about how electrons are moving. Which way? Are you losing electrons or are you gaining electrons? Right? So oxidation is losing electrons. When something is oxidized, right, you can see this molecule, it has these two little dots, these two little electrons right here. It goes through oxidation and it's lost those electrons. Reduction is gaining electrons. When you reduce something, which is confusing for folks because you think reducing, oh, if I reduce something, I've lost something. So reduced molecules are losing electrons. No. What it refers to is reduction in charge. So you have to remember that electrons have a negative charge. And so when a molecule is gaining electrons, its charge has been reduced. It's been made more negative. Okay? Oxidation, when things oxidize, they break apart. They're breaking down. When you see a copper a bird house or bird feeder, or someone leaves their bike outside and it starts to rust, those things are oxidizing. They're breaking apart. They're breaking down. And when you break bonds, you're losing electrons, right? You're releasing electrons. So something that is oxidized has lost its electrons. The phrase, there's several kind of phrases. Again, your soft talk has these. Oil rig is a common one. That's one that I always remember hearing. Oil oxidation is losing electrons. Rig reduction is gaining electrons. The other common one is Leo-Gur, losing electrons, oxidation, gaining electrons, reduction. So oil rig and Leo-Gur, however you want to. This one, the book is showing this image is the people giving and receiving gifts. So the one that gives the electrons, that's the oxidized molecule. The one that receives is the one that's been reduced. And this is kind of how we start to create ions. When you think about ionic bonds, you're not sharing electrons, you're donating, you're taking and you're giving electrons. And so in this case, this molecule here, the one that has donated its electrons, it's actually going to be a little more positive because it's lost those electrons. So let's think about this as like a sodium chloride reaction, right? So if sodium is always a little positive because it's going to give its electrons to chloride, and then chloride becomes a little negative because it's gained sodium's electron. So you should be able to kind of think about, well, what was oxidized, what was in a sodium chloride molecule, which one is being oxidized, which one's being reduced. You should be able to identify that. These come into play with our conversation on energy because oxidation reduction, again, the process of transferring energy can come down to breaking and making bonds, and bonds are all about electrons. So how you transfer your electrons is going to kind of dictate how energy is transferred. Okay, energy moves with electrons. Kind of think about that. Energy moves with electrons. That's what batteries are. When you think about like a lithium-ion battery, lithium is an element. So they've ionized lithium with positive and negative charges, and with positive charges, and then you plug it in, you excite those electrons, and then they can flow down their current when you plug them into an electrical system and you complete that circuit. And so it's all a series of just moving electrons from one thing to the next, to the next, to the next. And as you move electrons down, you release a little bit of energy at a time. And so that's what you're seeing here. What we're seeing here in this image is an electron transport chain. So oxidation reduction reactions, we call them redox reactions for short. They always have to occur in pairs. You can't just have one thing being oxidized. If you oxidize something, you have to reduce something else. If you break one bond, you're forming it somewhere else. And so biological systems have evolved to be able to really harness this effectively. And so what they do is they take a stretch of the plasma membrane. So they'll take a stretch of membrane and they'll put all of these proteins on here. And so each of these proteins are part of what we call an electron transport chain, an ETC. And what happens is you start with this a molecule that has some electrons that it's ready to donate, it's ready to get rid of. And then these molecules are very electronegative. Each one is slightly more electronegative than the last. And so this molecule, it's ready to get rid of its electrons, right? It's happy to be oxidized, happy to donate. Oxidation is losing electrons. And so as this one is oxidized, this first molecule gets reduced. It's gained its electron. Well, this molecule right here is more electronegative than this first one. And so this first molecule now, this first protein, is going to donate its electron that it just gained. So now it becomes oxidized and this protein is reduced. And so on and so on and so on, each molecule being slightly more electronegative until you kind of get to this very, very end one. And along the way, each time you move that electron, you release just a little bit of energy. And electron transport chains, you'll see, will harness this. They'll add, right? We're only seeing the electron transport proteins, but this protein right here, because you see how it's kind of actually two put together, this is actually probably like a hydrogen pump. So you're transferring electrons from one to the next. And as you transfer it, this energy that's released can actually fuel a hydrogen pump. Or some other kind of enzyme that's going to make a reaction happen, right? And so that's how we harness electrons, harness these this kinetic energy to do work, because this is kinetic energy, right? You're moving electrons from one thing to the next, you're releasing energy. These are kinetic reactions. We're not storing the electron in one of these. If we were storing and leaving that electron in one of these molecules, that would be potential energy, but it's being transferred over and over, releasing energy. So these are kinetic reactions. Okay. So if you look at this, right, just give you a quick second. In this reaction at the end of the electron transport chain, molecule X is being what and molecule Y is being what. So you have, let's see if I can zoom in here. There we go. You have this final protein here in the chain and this last thing that's being accepted. So it's trying to ask you, and the answer is right there on the screen if you can read, is molecule X, is this protein, as it gets rid of its electron, is it being oxidized or is it being reduced? And if you remember, this molecule is losing its electron, so it is being oxidized, while this one is gaining its electron, it is being reduced. So, yes. Okay, skip that. Cool. So where we're going to see this come into play, and when we talk about the energy of the cell, I mentioned glucose, I mentioned electrons. Really, the big energy molecule is called ATP, adenosine triphosphate. So ATP is cellular energy currency. When someone talks about their blood sugar being low, when someone talks about needing, getting a sugar high, which by the way, a sugar high is not a thing. Sugar does not give you a spike in your energy. Caffeine causes your energy to spike. Sugar doesn't cause your energy to spike. Typically, when people are complaining about kids getting a sugar rush and having all this wild energy, it's like at a birthday party or at a holiday celebration where they're probably getting presents and they're really excited anyway. Sugar doesn't give you a rush of energy. It gives you available sources to make energy, but caffeine is the thing that makes you jittery. Like if you're giving your kids soda, yeah, that's gonna make them all kind of jacked up. But just like cake and ice cream, it's just sugar. Your body, if it's not using it, then it's just gonna convert it to triglycerides and glycogen and store it as fat. But if it is able to use that energy, if let's say you are running a marathon or a half marathon and you carb loaded the night before, you ate a bunch of pasta, and so your body has all of this glucose, it's going to take that glucose and it's going to convert it to this molecule called ATP. It's kind of like if you go to another country, you can't just take your American dollar bill, even if you're going to Canada or Mexico, right? You have to actually convert your money into a usable form that's there. And so that's what the cells do. We eat food. Our bodies break that down into the small parts. It takes the glucose molecules that it's able to, that it has, it makes them if it needs to, and it transports those to the cells. Cells then take glucose, because glucose is a pretty big molecule and it's very stable. Glucose doesn't want to be broken apart necessarily. It's pretty happy how it is. And so it takes a little bit of effort to break glucose apart. But once glucose gets broken down, one glucose molecule makes about 36 ATP molecules. So you can really, you don't need all that glucose. It's kind of like, you don't need all that glucose, right? You only need a couple of ATP molecules to do things really. And so ATP is a usable form of energy in the body. Glucose is usable too. We'll get to it. All cells, though, rely on ATP primarily. So ATP is this adenine nucleotide. If you remember from your RNA, DNA structures, your nucleotides, you have an adenine nucleotide over here. You have a ribose sugar right here in the middle, and then you have these 3 phosphate groups. And these little squiggly lines right here represent high energy bonds. Rather than like a solid straight line, it's this kind of little squiggly line, and that means that it can be broken pretty easily. And so that's where ATP kind of comes in as an energy player, is that these bonds can be broken, and when you break a bond, as we saw with oxidation reduction reactions, you're releasing that energy. Those electrons get released, and that energy gets released. So ATP, like I said, gets formed during cellular respiration. It's a series of reactions in the mitochondria, right? You take food, take something that you've eaten, your body is going to break it down, break down that thing, and as it does, it can make ATP in the process and a little bit of water. It's called phosphorylating, by the way, when you add a phosphate, because we start with an ADP, adenine diphosphate, 2 phosphates. We need a triphosphate. And so when you add a phosphate group to something, it's called phosphorylation. So that is ATP production. ATP hydrolysis, breaking open ATP, again, you're releasing that 3rd phosphate, and in doing so, releasing energy, and then that energy can be used to make and fuel reactions. So the hydrolysis, the breaking of ATP, gets coupled with endergonic reactions. So if you remember an endergonic reaction, you're putting energy into a system. Well, that energy comes from breaking ATP. So we break open an ATP, or we break apart that 3rd phosphate, and as you break apart that 3rd phosphate, the energy that gets released from it can then be used directly to do some kind of work, to do some kind of job. It can be something like adding a phosphate. That sometimes is all that it is. It's literally just taking that phosphate that was broken off, attaching it to something else. Adding phosphates to things, it's kind of like turning them on or turning them off. It activates and deactivates depending on the molecule in the process. So glucose here can be activated by a phosphate. Activated means that it's more likely to bond. And so now this 1 glucose monomer, because it's been activated, is more likely to bond in this little polymer chain. And when it does, it's now added to this polysaccharide, the phosphate is released, and this phosphate can then be added back to an ATP. You can reattach it. It's reusable. So it's a really efficient process in that sense. Okay. Here's another way. You can see this right here is a cell pump, it's a transport pump. So sometimes you need to get stuff, if you remember the discussion of the plasma membrane, some things can cross, some things can't. Ions have a hard trouble, charged molecules have a hard time crossing the plasma membrane. And so sorry, I'm just making sure the testing center isn't calling me. Some of y'all are still doing your exam, and so I want to make sure I'm available. Okay, so you see that you have molecules on the outside, and you want to kind of get them to the inside. Well, they can cross through a transport pump. The original protein shape, right? You see that it's kind of open to the inside. It's got these molecules bound in here. Once this ATP binds, it adds its phosphate. That phosphate is going to change the shape of the transport pump, and then it's going to be able to open it up. So when we talk about energy being released, sometimes it is just the energy that is then fueling the reaction. Sometimes we're adding a phosphate. Sometimes it's just about adding and removing phosphates when you're talking about using ATP. It's a very helpful molecule. Jumping into enzymes. Okay. So a lot of the reactions in a cell, when we talk about things happening in a cell, and obviously that's the functional part of life, is occurring through enzymes. Enzymes are there to help speed up our biological reactions. Without enzymes, we would not be able to exist. Life would not exist at a level without enzymes, because it's not that reactions wouldn't happen. That's always something that biologists like to make clear, is that these reactions would still happen, but they would happen at such a slow rate that we would not be able to sustain life at our level. We would have single-celled organisms, very, very small microcellular organisms. We wouldn't be able to manage large organisms like we have without enzymes. So the most important of all biological molecules, they are proteins. Enzymes are protein molecules. They help to catalyze reactions, and they are reusable. So once a reaction, when it's an enzyme catalyzes a reaction, it can then go on and catalyze that same reaction. So enzymes are proteins that act as catalysts. They speed up chemical reactions without being consumed. So they're reusable. Some terms we want to make sure we know: enzyme is the protein that's doing the thing. Enzymes bind to substrates. The substrate is the molecule that's going to fit the enzyme, and it's going to bind to its active site. And this is we call it very specific fit. The shape is very exact. And when we say shape, it's really molecular bonds, right? That's what's happening here. It's not like we're kindergarten and we're just matching block shapes. It's these molecular bonds are going to fit exactly. Each enzyme is only going to bind one substrate. You're going to have one enzyme that's going to catalyze. You might have millions of copies of that enzyme, but the enzyme is only going to catalyze one reaction. You're not going to have one enzyme that can catalyze multiple steps because this active site is so specific. So once the substrate binds into the enzyme's active site, we make what's called the enzyme substrate complex. This binding, this very kind of tight fit binding, actually changes the shapes. And when it changes the shapes, it doesn't quite fit as like it's supposed to, and so it releases its products. So the substrate becomes products. This is showing things breaking apart. Sometimes enzymes put things together, so it's always breaking things. But what you have to remember is that with these very small molecular structures, so much relies on the shape and the conformation of these molecules. If the shape changes, then the activity changes. Protein form and function are directly linked. So with an enzyme, or rather with the substrate, once it binds, the substrate is the correct shape to bind the enzyme. But once you form the enzyme substrate complex, it's no longer two separate molecules. It's now one new molecule, and this new molecule is actually not designed to fit in the active site. It's a very strange thing, but it's one of those, it's like a Theseus ship kind of conundrum. It's like, well, if you have a ship that's slowly breaking down, and as it breaks down, you replace each board along the way, so the ship never is really gone, but is it still the same ship at the end? In this case, it's not. Right? Once the substrate binds the enzyme and forms the enzyme substrate complex, this substrate is now no longer. It is now a new thing, and this new thing doesn't fit the active site because the only thing that fits the active site is the substrate. It's a weird thing. We can talk about it in office hours if you want to. But essentially, that's going to cause it to eject the products because it no longer fits. It no longer binds correctly to the active site. This is just going over what I was just saying. The only thing that I want to reiterate, right, is that the enzyme does not change shape. The substrate becomes the products. Those get ejected. The enzyme stays the same. It's going to then find more substrate to bind. Okay. They speed up a variety of reactions. Like I said, they're not always just about breaking things down. Sometimes they do help to put things together. When we're talking about polymerizing macromolecules, enzymes are really helpful with that. What they do is that they lower the energy that's needed in a reaction, what we call the activation energy. So while, yes, they may still require energy to happen, enzymes make sure things happen in a much, much lower energy need. So you can see this kind of graph over here. You have these two, this kind of purple and this goldish yellow. These walls are showing how much energy is required. So in this purple one, you see it's only a little amount of energy that's needed, whereas in this one, there's a lot of energy that's needed. And so this one here, this yellowish gold one, is uncatalyzed. So there's enzyme helping this reaction to proceed. And so you need a huge amount of energy that's almost insurmountable in the body and the cells. Whereas with an enzyme, oh, your body only needs to add a little bit more extra energy to get it to happen. And so, yeah, this is totally manageable. We can do that, right? This much, oh no, that's too much. But this little bit, right? Oh yeah, we can do that, no problem. So enzymes just help to lower the amount of energy, the activation energy that's needed. And again, it's because once that substrate binds, that's going to just kind of pre-break or pre-form. It just changes the shape enough of that substrate to make sure that these reactions can then proceed pretty quickly. Sometimes you have things called cofactors that help to catalyze reactions. They participate in that activity. They help to increase activity, ions, vitamins, those things. So this is one of those things where you can survive with just calories, right? Really, all you need are just calories. I always kind of talk about how when I was growing up, for middle school, every day for breakfast, I ate donuts and Mountain Dew. I loved it. That's what I have for breakfast every day. You eat multiple meals throughout the day, all throughout the week, so you can get your vitamins and minerals somewhere else. But you got to make sure you're doing that because for me, right, I was just doing donuts and Mountain Dew. There's really no vitamins or minerals in that. I had the energy. I was getting calories that my body could turn into glucose that my body could turn into ATP. But there are certain enzymes that need vitamins, your B12 vitamins, your B vitamins, your C, A, D, E, all those different things, magnesium, potassium, all those other things that you need as cofactors. And they help facilitate and speed up those reactions even more, or they make sure that it actually will happen. Without some of these cofactors, some of these won't happen. So this is why it's always good that, yeah, have what you want, but also include some varieties, some nutrient dense foods in your meals as well. So that way you're getting these cofactors that you need, right? This can be the difference between DNA replicating properly and DNA not really replicating properly. If you notice like new little spots that start to pop up on your skin, that's one of those signs, like, a lot of times, especially in Western and in more traditional Eastern medicines, like Ayurvedic medicine, the first thing they tend to look at is your skin because that's your largest organ. And so if you start having new spots and new things pop up on your skin, everybody knows, oh, that's something different. We need to look at that. That wound isn't healing. Why isn't that wound healing? Well, if you're not getting the right vitamins and minerals, your body is not optimally performing these enzymatic reactions, you're not going to recover properly. So enzymes are proteins that are just going to keep catalyzing reactions. If there is substrate around, enzyme will bind it. It is a mindless process. It'll just keep on happening. So cells have to have a way to limit and to inhibit that process. And so we can do what's called inhibition, competitive or non-competitive inhibition. Because only some reactions need to occur at a time. If you are not going through puberty, or if you are already outside of your puberty stage, your body doesn't really need to be producing that much testosterone or estrogen or whatever it may be. And so you can start to inhibit those processes, and you don't really need to be doing it, right? It's one of those things like, well, why is the 7-year-old producing so much testosterone? Let's go get it checked out. Something's not right. And so this enzyme inhibition prevents unneeded reactions from taking place. You can see here in a normal reaction, the substrate binds into the enzyme active site, all good. Well, in non-competitive, you have this inhibitor that binds somewhere else and changes the shape of the active site so the substrate can't bind. In competitive inhibition, you have your inhibitor binding right to the active site, blocking this substrate. So a couple different ways. This is how a lot of medications will work. So when you talk about medications that are for preventing HIV, like there are some medications that once someone has contracted HIV, they can take a medication that inhibits the replication cycle, or medications that are inhibiting a synthesis of molecules. If your body overproduces certain molecules, you can take an inhibitor that is going to essentially compete with that enzyme. So we call this non-competitive, this one we're looking at right here. We're calling this non-competitive inhibition because the inhibitor isn't actually competing with the substrate. The inhibitor does not bind the active site. And so if the inhibitor isn't binding in the active site, it's non-competitive. And this is because the inhibitor can bind what's called the allosteric site. And the allosteric site is a non-active site on an enzyme. And when the inhibitor binds, because again, forming chemical bonds, breaking chemical bonds, you're going to be changing the shape of the molecules. So when an inhibitor binds an allosteric site on an enzyme, it actually changes the shape of the whole enzyme. And so the active site, rather than being kind of this squarish, now becomes this round. And well, what do you know? That no longer fits the substrate. So the enzyme is inactive. As long as the inhibitor is not being broken down, it doesn't matter how much substrate your body is producing, doesn't matter how much of a molecule your body is producing, your body is not going to be able to break it down. SSRIs, selective serotonin reuptake inhibitors, they help to increase the amount of serotonin that's in your body to help treat depression. What's one of the things that's happening is that these inhibitors are preventing the enzyme from breaking down your serotonin. So you have this non-competitive inhibitor that changes the shape of your enzyme, so that way serotonin, that molecule, actually can't bind and can't be broken down. And so it means more serotonin in your nervous system, and hopefully less depression. So non-competitive inhibition does not bind to the active site, does not matter how much substrate is there. It will just keep binding until it gets broken down, because everything gets broken down eventually. That's why you have to constantly take medicines, or keep taking medicines on a regular basis as prescribed by your doctor. Competitive inhibition, then, so the other kind, it's competing for the active site. It's competing with the substrate. So the inhibitor has really similar enough shape to the substrate that it can also bind in the active site. And so a lot of times, again, medications might make a synthetic molecule, a synthetic substrate that rather than, or a synthetic inhibitor that mimics the shape of the substrate, but actually is not active. It does not release itself. It stays bound to the enzyme so that the substrate can't bind itself. So again, you can see inhibitor is competing here, which is why we can call it competitive inhibition. Competitive inhibition can be overcome if your body just keeps producing substrate, more and more substrate. Eventually, that substrate will outcompete the inhibitor, especially as inhibitor gets broken down. If you forget to take your medication as you're supposed to, blah, 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 that substrate will have more opportunity to bind. I will say with the non-competitive inhibition, once the inhibitor, right, and non-competitive, once the inhibitor is released, the enzyme will go back to its normal shape and be able to catalyze the reaction. Okay, so the body can use inhibitors, right? I keep talking about medications, but your body will also just make inhibitors. It does not have to be this medication that you're taking externally. And it's a very normal process. It's not like inhibiting enzymes means that you have some type of condition or disorder, right? It's actually a process of negative feedback. If you remember, we talked about negative feedback before. This is a way to control these biochemical reactions. So really, like, let's think about that, right? Like, if your body is producing a hormone, well, at a certain point, you're going to make enough of that hormone. And if you keep producing too much, you're going to over compensate. Your body's going to have an overreaction to it. So what happens is that hormone, once the final hormone that's produced, the final product that's produced, will actually go back and act as an inhibitor on that first enzyme in the process. Okay, so let's say you start with substrate A, right? You're starting here with substrate A. It binds to enzyme 1, it gets released and is now B. B binds to 2, and then blah, 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 so on and so forth, until you get this final product. Okay, this final product, it's not the same shape as any of these other substrates, so it can't bind competitively. It's going to go and bind over here, act as a non-competitive inhibitor on this first enzyme. So as product accumulates in an enzymatic chain, in a biochemical chain, the product acts as an inhibitor to the whole process. Eventually, right, as product builds up and it inhibits enough enzyme, you just stop producing that product. Well, once that product is no longer being produced, its levels are going to slowly drop. And once those levels drop, you won't have enough to inhibit the enzyme and complete the action that it needs to. Because remember, this product is supposed to be doing something. This product is supposed to be out doing a job, doing something. And so if you have enough product that it can all do its job and then some, can inhibit the reactions, okay, cool. Yeah, we don't need any more. But eventually, this product that's inhibiting these reactions, it's going to get called in. It's going to be like, hey, we need you to get out here. We actually don't have enough now. And so as this product unbinds, right, it kind of leaves these enzymes to do their thing. Well, they're going to start producing more product again. And so that's negative feedback, right? The body kind of trying to reach this set point, but always kind of moving slightly just off until it trying to reach at that set point though. It can also use positive feedback. I don't have a picture because the picture from the book is the same one as the negative feedback picture, and that just always confuses me. Just remember that positive feedback is moving towards something. The product actually stimulates its own production. It stimulates the production of more of itself. We see a lot, the big main things with positive feedback are labor and delivery and milk production. Those are the big positive feedback processes. Okay, so that's how the body can regulate enzyme activity through inhibition, through substrate activity. Other ways do also alter enzyme activity. They're very sensitive to temperature. So temperature is a really big way that our body will kind of fight infections and try to fight different enzymatic reactions. So in humans, body temperature is 37 degrees Celsius. And so our enzymes kind of function best right there, right at 37, 38, maybe 39 degrees Celsius. So actually, when you have a slight temperature, your body is actually functioning better. Your enzymes actually function better at a slightly higher temperature. But as soon as we hit about 41 degrees Celsius, our enzymes, you can see they just plummet. It's because they start breaking down. The protein shape actually starts to denature once it starts to get a little over 41 degrees Celsius. This is about 104 degrees Fahrenheit. And so 104, 105 degrees Fahrenheit is about the temperature where brain damage can start to occur. So if you're an adult who has a temperature, if you're a human that has a temperature for a period of time that is over 104, brain damage is a big concern because your proteins are going to start to break down. And you can see that again, enzymes are proteins. Bacterial enzymes, they have a much wider range. You can see they function at a much, much higher temperature, almost all the way up to 78-ish degrees Celsius. This is one of those things where, washing your hands with warm water is great, but warm water is not going to do nothing to those bacteria. If you aren't using soap, if you're not using some kind of detergent, you're not actually getting rid of any bacteria. You're probably just making it a nice little bath for them. That's why when you work in like a medical office, the steam sterilization, it has to get up to a certain temperature. That tape is only activated at a certain temperature. And so oh, okay, we got to the temperature we needed to because our tape was fully activated. And it needs to be there for a period of time. If you ever get like a boil warning, I grew up in a county where a lot of people had well water, we had a lot of bad county water. So we would get boil notices where, hey, if you're going to drink your water, make sure you boil it to this temperature for this period of time, for 10 to 15 minutes at this temperature, because it's not just the temperature, it's also the time. That length of time makes sure that you really destroy any enzymes or bacteria or organisms that might be dangerous. So temperature, very important in regulating enzyme activity. pH is also going to be a function, going to regulate enzyme activity. Remember, pH is a reflection of how many hydrogen ions are in a solution. So as your pH starts to go up and down, your hydrogen ions start to go up and down. That's going to affect hydrogen bonding. Hydrogen bonding is crucial for protein shape. Okay, so it's all coming back together. So if you start to adjust your pH again, your pH can adjust. It can only modify about 0.4, 0.5 points, like about half a point adjustment before your proteins start to denature. So very, very tightly controlled. Okay. So we're going to kind of shift gears here for just a little bit. Moving from a discussion of enzymes and those kind of reactions to membrane transport. I know it kind of seems odd, but all of this kind of relates to energy because enzymes are part of our chemical reactions, enzymes fuel reactions. And when we talk about membrane transport and substances moving in and out of the cell, a lot of that is involved with energetic processes. There's passive and active processes here. So just again, that's why I usually like to break this lecture up. So apologize for all kind of cramming this in here at once. We started this lecture with that conversation on the different kinds of energy, potential and kinetic energy. We talked about the different ways that we can look at it. We can look at it as metabolism, like building and breaking structures. We can look at it as endergonic, exergonic. Are you putting energy in or are you releasing energy? And we looked at it through electrons, those oxidation reduction reactions. Are electrons being gained or lost? We talked about electron transport chains. We talked about how transferring and moving electrons is a way to release a little bit of energy in field processes. And then we kind of got into that conversation with enzymes of actually catalyzing and fueling these reactions. This next part is about establishing what's called concentration gradients and how concentration gradients can be used to also fuel reactions. Okay, so we can use direct energy like glucose and ATP to fuel reactions. We can use enzymes to help catalyze and proceed with reactions. We can also use things called concentration gradients, and those can be used as a source of energy for reactions. Okay, so I guess that's a good way to kind of clump it all. It's like, okay, how are you accomplishing your reaction? Are you using ATP and glucose as your energy source? Are you using enzymes, or are you using concentration gradients? I like looking at it like that. So if you remember, the plasma membrane is made of this phospholipid bilayer. It's semi-permeable. It allows small, non-polar, non-charged substances in and out. But otherwise, it creates this barrier, creates this space inside and space outside. Intracellular inside, extracellular outside. So that regulation of what can and can't enter is part of homeostasis. Things that can flow freely, that's great for them, but other things require proteins to be able to cross in and out of the cell. And things tend to try to find balance in nature. That is just a natural, that is just a law of nature, that things are going to find balance. This does not have to necessarily be with a living organism either. We see it in all things. So here we're looking at a cup of tea, right? I'm drinking my tea as well. So if you ever drink tea, right, you have a little cup of water, you put your tea bag in there. If you just kind of let that tea bag sit, eventually the water will start to turn the same kind of brownish color because the tea molecules, the molecules here, are moving down their gradient. They're diffusing into the rest of the solvent, into the rest of the water, and evenly distributing. If you were to do something like heat the water up, dunk the tea bag a little bit, maybe stir it, that would help to speed up the rate of diffusion, and it would disperse evenly faster, right? But if you just let it sit there, it'll just naturally do that on its own. The tea bag will just naturally diffuse. The tea will just naturally kind of diffuse out. That is just a part of life. When I teach classes in person, when we do classes in person, never do I come into a room and everybody is, in a 50-person room. I don't have 20 people gathered all in the front in one quarter. People are naturally going to disperse all out because we don't things in life. Nature does not like to be crushed and pushed up next to itself. We want a little bit of space. Right, I often kind of get this. If there was a room, you walked into a room and there were 20 different organisms in the room, and 10 of them were humans and 10 of them were puppies, would you go sit in the group of 10 humans or the group of 10 puppies? You would probably wonder why nobody was sitting with the puppies, but you probably want to go sit with the puppies. You don't want to be in a group that's really tight with all the same kind of things. Naturally, we want a little bit of space, unless it's something a little different. Like I said, if it's some other things around us, yeah, we don't mind being crowded by other things. I've got a desk and papers and all this other stuff around me now, but I'm not crowded by it. I don't feel crunched. But if I had 20 people in this room with me, I would feel really crunched and trying to find space. Everything naturally will move down its concentration gradient. Go and look like it's you like city planners use this kind of stuff a lot to talk about like how traffic naturally flows, how crowds naturally flow. They use these same kind of dynamics. Also, I know it's about to be 3 o'clock. This one will definitely go a little bit later. So if you need to log off, that's fine. I'm just going to keep on jogging through. Okay, so gradients describe a difference between neighboring regions, and cell membranes allow us to establish that gradient. So gradients are directional. Things are going to move down their concentration gradient. Get that into your brain. Nature will naturally passively move down its concentration gradient, meaning you start at an area of high concentration and go to low concentration. Okay, this T, it starts really highly concentrated in one area until the molecules have moved out. They kind of hit up next to each other and they kind of push against each other until they've all kind of got their space and like, okay, now we're kind of good, right? The gradient it dissipates. Example of I'm not reading all that. Okay. Typically, though, maintaining a gradient requires energy input. I know you're like, no, it doesn't. If you were to just let this T sit for a day or 2 days, it would start to separate. The T molecules would eventually, because of their density, would all start to separate. So while you have to remember with science, while I'm teaching you one concept about concentration gradients, there's also some other chemistry, some other physics, some other things that are happening that are related as well. So without a little bit of constant kind of like stirring, right, without a little bit of agitation, the T molecules will eventually start to settle and the water will start to separate. And so it takes a little bit of energy to keep that gradient up. Yeah. Okay, so crossing a cell membrane then happens in several ways. So trying to go down in that gradient, you can either do simple diffusion. Well, sorry. Let me start back. When you're trying to move something across a membrane, you are either going to be moving it down its gradient or against its gradient. Okay, so if you're moving something down its gradient, that is a passive process. That means it's going from high to low concentration. Things like simple diffusion, osmosis, and facilitated diffusion. These are your three passive processes. If you're trying to move it against, we call that active transport. Things like endocytosis and exocytosis. We'll see active transport. Typically, you're going to be talking about specific proteins rather than general processes. Active transport, again, is characterized by, well, it's going against its concentration gradient. You're moving something from an area of low concentration to an area where it's already highly concentrated. So starting with the first passive process, simple diffusion. So you can see here these molecules, there's an area of high concentration. They are passively just going to move, and these are able to move through the cell membrane. You see no transport protein that's helping them. So that's why we call it simple diffusion. Two things that word tells you. 2 words, 2 definitions. Simple means that there's no protein helping it. Simple means that this molecule, these little balls, are able to just flow right through the plasma membrane without any protein assistance. Diffusion is telling you that it's going down its concentration gradient, that it's a passive process. So simple diffusion. Simple, no transport protein needed. These might be oxygen molecules, right? These might be oxygen and carbon dioxide, who knows what. So they're able to just move right through that plasma membrane. Simple diffusion, going from high to low. Oh yeah, that's diffusion. That's a passive process. Again, because of their size, because of their charge, that's why they don't need a transport protein. That's why we call it kind of simple diffusion. Another passive process is osmosis. Osmosis specifically refers to water, so not any other kind of molecule. We're looking specifically at water, and it's important because sometimes water doesn't always move. Or rather, actually, you have to remember that things are going to move down their concentration gradient. So even if you have a substance, a solution where you have, for instance, this one, where you have a lot of molecules on one side and not a lot on the other, these molecules are going to move down their gradient. They're going to be going into this area of lower concentration. But what you have to think about is that in an aqueous environment, well, water molecules are less concentrated here than they are here. So while these molecules are moving down their gradient, water will be going the opposite direction because that is actually down its gradient. So each molecule is going to move down its own concentration gradient independent of other ones. And those things might be going against each other or with each other in the same direction. It really depends. That's why we talk about diffusion refers to solutes. Osmosis refers to water movement. So in this a picture right here, you're seeing a tube, and that tube, the two sides are divided by the semi-permeable membrane, representing a cell membrane. You have a lot of water molecules on this side. You have not as many water molecules, but a lot more solutes on this side. 1, 2, 3, 4, 5 in this space that we can see compared to 2 over here. So in this space, the solutes are going to want to move down there. So the solutes are going to want to move into this left-hand side, but the water is going to want to move into the right-hand side. So the thing that can happen, though, is that if this semi-permeable membrane, it's not actually going to allow solids to move. It actually looks like it's too small for these large solutes to get through. So rather than the solutes moving to the left, you're just going to have water moving to the right until it reaches a relative equilibrium. Okay, so again, in our original starting point, the volume on the two sides are equal, but the solute concentration isn't. One side, this right side, has a higher solute concentration. Over time, the solute concentrations are going to equalize between the right and the left sides, but the volumes are not. And that's an important thing to remember with biological systems, is that they're not concerned with getting to equal volume. They want to get to equal concentration. So it's okay. Well, we're going to see there's some conditions where it's not okay. But it is absolutely plausible and natural and possible, and very much happens all the time, that you're going to have, in an effort to establish equilibrium between solute concentrations, you're going to have one side that's going to be gaining water while the other side loses water, to make them relatively equal in solute concentrations. And again, that's still things moving down their concentration gradient. So how is this relevant? Where does this become relevant? We look at red blood cells. So what's important to remember is that water always moves to the hypertonic space. Plug that into your brain, get it stuck in there. Water moves to the hypertonic space. Whatever compartment has more solute, that's where water is going to go. It's going to flow out to there. So a red blood cell, and an isotonic, we have 3 different terms here. We have isotonic, hypotonic, and hypertonic. These are ways that we describe different solutions. Okay, so solution concentration. I'm going to actually flip back to this slide real quick. If you look at this one, you have a lot of solutes in this area and not a lot of solutes in this area. So it's unequal. So over here, the area with a high concentration of solutes, we would call this the hypertonic. And in this one, the hypotonic. Okay, they're not equal. One has more hypertonic space. The other one has less, the hypotonic space. What's important to remember is that you can't use these terms and only talk about one thing. I can't just say, oh yeah, this, you can't even see it. Here you go, let's see if this will show up. Okay, here's my glass of, this is my jug of water. I can't say, oh, this is hypertonic. Hypertonic to what? You have to describe it relative to something else. So I can talk about, in this image, I can talk about this side, this high concentration being the hypertonic space, this low concentration being the hypotonic space. But I can't just describe one space as hypotonic, hypertonic, isotonic. Okay? You have to be comparing it to another solution, comparing it to some other kind of space. So looking at red blood cells. This red blood cell right here, we're saying this is a blood cell in an isotonic solution. An isotonic solution means that the two sides have equal concentration of solutes. When you go to the doctor and they give you an IV if you're low on your blood fluids, and they give you an IV, they're giving you saline solution. They don't just give you pure water. They give you saline solution, has some solutes in there that's going to match your blood volume, your blood plasma. So that way it's essentially going to look like the same. So you don't have any big shifts in water, right? Equal solute concentrations. So water in and water out. Water's still going to move, but it's going to be moving at an equal rate. And that's important for maintaining blood pressure, blood volume, and proper cell shape. If you, let's say, have a hypotonic solution, this would mean, this would be something like if you were out running a marathon, right? Let's say you go out and run a marathon, or you're working hard in the yard. Actually, you know what? Here's a great example. I have a friend, and my friend loves to do hot yoga, and she does hot yoga so many times a week, and she is not good about her recovery, and so she really sweats out a lot. She uses a lot of her solutes. In a hot yoga class, you're sweating a lot, and when you sweat, you lose water, but you lose at a higher rate things like salts and things like that. And she tends to just drink pure water to recover. She doesn't eat after. She just drinks pure water. Well, when you do that, you're actually diluting your blood. It's actually turning your blood into a hypotonic solution. Your blood doesn't have enough solutes. And so what it thinks is that your red blood cells now become hypertonic. And so water rushes in, right? Water moves to the hypertonic space. If you chug a bunch of just plain water, this is when like if you're like, they don't do it as much anymore, but it used to be very popular with like radio contests and like frat hazing things where they would just make people chug a gallon of water or chug a gallon of milk. When you do that, you actually dilute your blood to a degree that water is going to rush from your blood vessels into your cells, and it's going to cause your cells to explode. It's called lysing. Your cells are just going to explode. Your brain can explode. Your brain can start to swell. You get cerebral edema. You can start bursting blood vessels, or you start bursting cells in your brain. It is an absolute life emergency. Hypotonic hydration. And again, it's when you drink too much plain water and you dilute your blood so that your cells become hypertonic and water rushes in. The other kind is hypertonic. So this is like if you get dehydrated and you're not like recovering, you're not drinking anything. You go to run a marathon, and instead of hydrating properly as you run, you just keep running, you just keep sweating and sweating. Well, when you do that, you lose water and you lose solutes and things like that. Well, your body is going to replace the blood in your veins from the water in your cells. So the water in your cells is going to start leaching out. So when you hear someone being dehydrated, they pass out from dehydration. What dehydration actually is that your cells have lost all of their water and are shriveling up, and water is moving out of them at a really high rate because the blood is hypertonic. The blood doesn't have as much water, and so it appears to have more solutes. So that's where osmosis and tonicity, how water moves, is really relevant to, well, if we look at the hypertonic solution, water always moves to the hypertonic space. In biological systems, you want to always make sure you're using isotonic solutions so you don't have any fluid imbalances occurring. It's also important for plants. Osmosis determines water content in plants. If you see a plant kind of that's starting to wilt, maybe it's placed in hypertonic surroundings, the water has been leaching out of its vacuoles. Whereas if you have water that is in hypotonic surroundings, the water is going to be rushing into those vacuoles. It's going to keep them nice and full, keep the leaves open, and as the leaves are nice and open, they can absorb sunlight at a higher rate. We call this turgor pressure. Plants say turgid, or the turgor pressure, turgor pressure. I don't know how you say it, but that's what prevents plants from wilting. So that's why when it's always, I'm one of those kind of people, like I like to watch plants kind of like wilt and then water them and watch them like reinflate in a day. And it's because the cell walls keep the structure. You just have to fill up the vacuoles to make them open. Okay, so that's water movement, right? So still a passive process. Water is going to be moving down its concentration gradient passively. The third type that we talked about is facilitated diffusion. So again, simple diffusion, simple, no protein required. Diffusion is moving high to low. Facilitated diffusion means that it needs help. If you facilitate something, you're helping it along. So facilitated diffusion going from high to low, so it's going down its concentration gradient. Facilitated just means it has its protein here. So it's something charged, something large, something that does not move naturally through the plasma membrane. Maybe it's a polar molecule, right? You're going to have a proteins for that. Facilitated diffusion is what makes ATP. You have these slides. You can look at this video. It's a YouTube video that kind of shows how this synthase pump works. So here you can see you have a plasma membrane with a high concentration of hydrogen ions. That's what these little gray balls are. These gray balls are hydrogen ions. They're going to move down their gradient 3 at a time. They're going to move down their gradient through this facilitated pump. And what happens is that as they move through this pump, they're binding and releasing, binding and releasing. Each time they do that, they actually change the shape because that's, again, chemical bonds, breaking and forming bonds means that you're changing the shape every time. And each time it does that, it actually causes that little part under there to rotate. And that thing will rotate 3 times. And 1, 2, 3, it'll eject the hydrogens. And in doing so, it phosphorylates ADP to ATP. So it's pretty efficient. This is called an ATP synthase pump. So this is an enzyme pump. We're going to see this a lot. We're going to talk about it in photosynthesis. We're going to talk about it in cellular respiration. So yeah, just be aware of ATP synthase pumps. You have high concentration, and we can harness that that concentration gradient. Again, this is all about how you fuel your energetic processes. Hydrogen concentration gradient, high gradient here. As it moves down its gradient, things moving down their gradients can actually then fuel energetic reactions. An example of active transport then, so where you are moving things against their concentration gradient. Remember that the big characteristic of active transport is, or if you're trying to decide, well, is this passive or is this active? Active transport is moving things against their concentration gradient. You don't need energy to move things down their gradient. Okay. So here you can see a sodium potassium pump. Sodium potassium pumps are absolutely crucial in all biological systems. They help to maintain that proper distribution of these ions for electrical activity. So outside of the cell, you have all this sodium. Inside of the cell, you have more potassium. And the cell is trying to pump this sodium out. It's trying to actually, you can see these 3 sodium, it's trying to move them outside of the cell. Well, to do that, it's going to have this ATP molecule bind that adds its phosphate. It changes the shape of the pump, and so it can be ejected. Once it opens up, these 2 potassium that are out here, they can then bind to the pump. That binding is a passive process. That's just a natural chemical process. Once they bind, though, their binding changes the shape enough that this phosphate releases. Well, this phosphate is what was keeping this protein open to that side. So once this phosphate opens, it reverts back to its normal shape. And then when it's in its normal shape, sodium can bind again. So sodium potassium pumps always exchange 3 sodium for 2 potassium. You have 3 sodium going out and 2 potassium coming in. Okay. Someone, a faculty member helped me, something that I liked was they said S out, P in, salt pin, sodium out, potassium in. Salt pin. Salt out has 3 letters, sodium out, 3 sodium. Potassium in, IN, has 2 letters, so it's 2 potassium in. I don't know if it helps or not. Another active process is called exocytosis. So again, active processes, moving things against their concentration gradient. Exocytosis, and the next one we're going to see endocytosis. This is when you're trying to move like really large things or a lot of something. You actually create these vesicles. You pinch off a piece of the cell membrane, or you kind of create it from like the Golgi apparatus or whatever it may be. You create this vesicle, this transport little Ziploc bag of phospholipids. Inside of it, you have the substance that's going to be expelled. Okay, the vesicles can bind to the cell membrane, and as they do that, they actually fuse the vesicle itself. The phospholipids of the vesicle become part of the cell membrane, and the cell just gets a little bit bigger, and then that allows these things to then move outside and push them outside. Endocytosis is essentially just the opposite. What's going to happen is that the cell membrane, instead of kind of staying flat, it'll actually start to like, I'm gonna try to do it this way. It'll actually start to reach up and around, and it will kind of send out these little processes that engulf whatever it wants, and then bring that in, and then it can pinch off and actually create a vesicle. So let's say that these are something that needs to be broken down. If it's just nutrients, that's no big deal. But if it were something toxic, you wouldn't want to bring something toxic into the cell and just let it be floating around. So by creating a vesicle with that, it actually keeps that substance isolated. So you can add things in there. You can add lysosomal enzymes or peroxisomal enzymes into this vesicle and just keep breaking things down kind of in a safe, neutral environment. But again, these things take energy to be able to fuse large structures like this with the cell membrane. All right. And that's pretty much what I had. So for this chapter, I really encourage you going into the study resources. There is a membrane transport flowchart. It's kind of got blanks in there, but it has explanations as kind of definitions for each of the different types of membrane transport we were talking about. So I would go in there and I would try to fill that out as best as you can. You're welcome to email it to me, and I can go over with you or meet me during office hours and we can talk about it. But I think that's a really helpful way for trying to organize membrane transport. Once again, for this chapter, it's all about energy. So, looking at the different ways, the different types of energy reactions, the ways that we look at energy. You have kinetic and potential energy. You can talk about reactions in terms of the structural changes, anabolic, catabolic. You can talk about it in terms of putting energy in or taking energy out, endergonic, exergonic. You can talk about the electron movement, oxidation, losing electrons, reduction, gaining electrons. Right? So all the different ways we can look at those energetic reactions. We talked about enzymes as catalysts for fueling those reactions, how enzymes are required to fuel a lot of reactions, for a lot of reactions to take place. And then we talked about concentration gradients and how active and passive transport can be used, either requiring energy or passive transport can be used to fuel some other processes. So this conversation, again, was a lot of theoretical stuff. Chapters 5 and 6, we're going to start applying these concepts, talking about photosynthesis and cellular respiration, and then really trying to give better kind of concrete examples of endergonic, exergonic, anabolic, catabolic. All right, if you have any questions, reach out, let me know. Thanks.